for attending the talk. And it's a real pleasure. I hope to visit Australia again soon. I loved it there in my very short visit. And today I will talk, we, as Fernando said briefly, we work on many different things in the lab. Today I will discuss the main topic that we study, which is epigenetic inheritance, transgenerational epigenetic inheritance. But I want to start very broadly, and in general, I won't go too much into the details because I know that people are coming from very diverse fields, but you are welcome at any time to ask me in the chat or to ask me after the talk about more details, and I'll be happy to expand and explain more. So I will start just by discussing the possibility even that memories can be inherited across generation. That's the title of my talk, and it's very provocative, I think, because we know intuitively that it shouldn't happen. We shouldn't inherit memories. Still, there is evidence that we are not born a completely blank slate. Some memories are encoded in the heritable materials, not necessarily the genetic materials, but that some memories are innate. And this is something that was already discussed by the Greeks thousands of years ago. There's also anecdotal evidence from more modern times. So, for example, experiments that were done in the 80s in the United States, and this is still in the realm of psychology, experiments that were done using monkeys that were born in captivity. So these monkeys don't know anything aside from what the researchers showed them, show that it's very hard to teach these naive monkeys that a toy flower is scary. So if you present them a toy flower and pair it with something scary like an electric shock or something like that, or a gong or something like this, eventually they'll understand that the flower represents something scary, but you will need to pair it with an electric shock for multiple times until they learn. Really. On the other hand, while the monkeys are not born afraid of snakes, it's very easy to teach them that snakes are scary. So it's enough to just pair it to something scary once so that they will remember it for the rest of their lives. So they are born with the tendency to be afraid, to learn to be afraid from snakes, but not flowers. The question is, where is this memory encoded in the genetic materials? We are all born from a single cell, a fertilized egg. So it's not in the synapses. So where is this memory? How is it encoded molecularly? This is a very difficult question and very interesting one, and I think it's a good angle to studying memory. So our brains, as you know, are extremely complex, so it's difficult to study in humans, which is why in my lab we mostly focus on the brains of C. elegans, or the nervous system of C. elegans, which is a very simple nematode, a worm, round worm, that has just 302 neurons, and it's always 302. And altogether, it has 7,000 synapses or less, which is the equivalent of what one neuron in human has. So it's really much simpler than the human brain or the mice's brain. And while the worm is very, very simple in comparison, it can still do many complicated things. So while it cannot write poetry or a tweet or things like that, it is capable of different behaviors. It can smell thousands of different odors. It can mate, it can sense touch, and it can even learn certain things, like very simple associations between food and odor. Most of what the worms need to do is find food. And so when I went to do my postdoc, that was 2010, I went to Oliver Hobart's lab, and I wanted to study how memory is encoded in C. elegans. That was my dream. I thought this is a good model system to study this in. And I wanted to focus on one particular memory, which is even a simpler memory. Just before that, I'll say that not only is the nervous system of the worm extremely simple, so it has just 302 neurons, we also know about all the connections. So we have a connectome, an entire deciphered connectome of the worm. We know with which neuron each neuron speaks and how many synapses each neuron form with other neurons. So it's like a subway map. And we have this map from the 80s. So for many years, people have been studying this connectome. And I wanted to study memory in C. elegans and focus on a particular memory, which is called imprinting. And imprinting is a good memory to start from because it's very well defined. It's a long-term memory that you can acquire at a particular developmental stage, usually early. So you're probably familiar with some examples of imprinting. For example, salmon swim against the current to the place where they were born. And a baby is imprinted, there's a neonatal attachment that is formed, a very strong bond between the baby and the mother. And this was discovered and studied by Konrad Lorenz, who got the Nobel Prize for that, who studied birds. And he showed that the birds will, once they hatch, they will track the first moving stimuli that they see. And this it's normally their mother, but as you can see in this clip, it can also be other things, a dog or a kid and so on. And also in C. elegans, in the lab where I went to do my postdoc a few years before I came, they discovered that worms can be imprinted with so that they can pair a neutral odor with food. And if they do this pairing at a critical stage during development, at the first larval stage of the worm passes through 4 larval stages, then the memory will stay intact, it will stay, and they will still be attracted to the odor even a few days later. Normally, when you pair odor with food in C. elegans, the memory just stays for 2 hours, so very short. But if you do this pairing at the particular developmental stage, at the L1 stage, then the worm remembers for multiple days. And this is quite amazing. And I thought, wow, this is an amazing paradigm for long-term memory. And indeed, in the science paper, they also showed that the memory is dependent on one neuron called AI1. It's an interneuron, and one receptor, SRY11. So it's quite an amazing level of description for a memory. So I wanted to study this. However, when I got there in 2010, so 5 years after this paper, my plans changed completely, because the first author of this paper, you see this paper is from Remy and Hobert. I went to Hobert's lab, and Remy is a researcher from France. And when they work separately. And when I got to Oliver's lab, Remy published a much crazier paper, even much more exciting, claiming that the memory is not only maintained in the animals that are paired, that learn the pairing of the food to the odor, but it also transfers to the next generation for multiple generations. This was really crazy, and this is just a sign for the craziness, because you know intuitively that this shouldn't happen. What I'm telling you now, if you'll remember it, it won't pass to your kids. It's obvious. Not only is it obvious, it's also, this possibility goes against one of the most fundamental dogmas of biology. It's called the second law of biology, or the Weismann barrier. And this is the principle that was thought of or formulated by August Weismann, one of the most prominent biologists working in the 19th century, who followed Darwin. And what Weismann said, he didn't use these words because these words didn't exist. But what he said basically is that the soma is segregated from the germline. Only the germline tissues, the sperm and the egg, can transfer information to the next generation. What happens in the soma stays in the soma. This includes also the brain, the nervous system. And Weismann, and we know this is true because only the DNA of the germline matters. And so this is why it's such a basic dogma. And Weismann formulated this law of his to show that Lamarckian inheritance or inheritance of acquired traits can't happen. Okay, so if you go to the gym, you build up muscles, your kids won't be stronger as a result. If you learn something, since your brain is disconnected or segregated from the germline, then it doesn't matter for the next generations. And this is a basic dogma, very fundamental in biology. The reason it's so well accepted, it goes well with what we know about genetic inheritance. So if you get a mutation in the DNA of your hand, it doesn't matter for the next generation, because the DNA of the germline, the sperm and the egg, is unaffected. And since only the germ cells will develop to generate the next generation, it doesn't matter what happens to the genome of your body. And this is also true when it comes to epigenetic information. So even chromatin modifications, cytosine methylations, or histomodifications in the germ cells are largely erased in 2 waves of reprogramming in the germline and early embryo. So most of the marks are removed so that each generation can start as a blank slate and develop according to the species' typical instructions, developmental instructions. And this is largely true, however, not completely true, because it's only about 90% of the marks that are erased. And perhaps the 10% that remain still could be very important. And there are many people studying this. However, by and large, there's a widespread reprogramming of most chromatin marks. Okay. So can memories transfer if there are all these barriers? Can memories nevertheless transfer to the next generation? That's what I wanted to study. And my starting point was that in C. elegans, when it comes to RNA, it seems like these worms don't care much about the Weismann barrier. We know this for 20 years. So we know this since the paper that awarded Mello and Fire with the Nobel Prize for the mechanism of RNA interference or gene silencing. In this paper, where Fire and Mello discovered that double-strand RNA is the substrate for RNAi, the double-strand RNA is being chopped up to small RNAs, leading to silencing, to gene silencing. In that paper, in addition to finding this important thing, they also found that RNAi moves between different tissues in C. elegans. It acts non-cell autonomously. So if you inject somatic cells of the worm with double-strand RNA, the silencing would spread to other tissues, including the germline. And moreover, if you just feed worms with bacteria, the worms eat bacteria in the lab. If you just feed them with E. coli that express double-strand RNA, the RNA that this double-strand RNA induces would move from the site of ingestion from the gut to the rest of the body, including to the germline. So you would see silencing also in the next generations. And this is something that every C. elegans lab uses routinely when we do RNA. No one scores the silencing in the mother. We score the silencing in the progeny because we want to see the silencing in hundreds of progeny. So this is extremely replicable. Now, in these papers, they showed very convincingly that RNAi spreads between tissues and also breaches the Weismann barrier. However, and over the years, we learned also how it works. We don't know everything, but we know quite a lot. We know about at least 8 different genes that function specifically in spreading silencing between tissues. So for example, there's the famous SID1 receptor, which a transmembrane channel which moves double-strand RNA between tissues. That's also conserved in humans even. And there are other genes. However, all of this is true only when it comes, or at least we know it's true only when it comes for exogenous artificial double-strand RNA. This definitely leads to non-cell autonomous silencing, but it's unclear. No one knows if endogenous small RNAs, natural small RNAs also move between tissues. There are hints that they do, and I will describe these hints, but we don't know which type of small RNAs actually move between cells. And when it comes to endogenous small RNAs, there's still a lot to discover and it's still a mystery. Okay, that's important to remember. So we knew all of that since 1998. And when I started my postdoc, I wanted to know whether there's any physiological relevance to this, whether actually this means something in the real world. Can natural RNAs transmit memory or parental responses, gene expression changes to the next generations? And if so, can these small RNAs mediate transfer of memory between generations? So what I did was I started reading about the possible connections between memory and RNAi and small RNAs. And I almost gave up because when I started my postdoc, there wasn't anything in the literature, nothing. So what I did was, I think you do it as well probably, is I just, after I gave up on the professional literature, I just looked in Wikipedia. And in Wikipedia, in the part about the ecology of the worm, there was this sentence that was since then removed. What this sentence said was that C. elegans is one of the few forms of life not known to have a natural virus. Okay, and this is very weird. The world, everyone knows now that viruses are extremely efficient at infecting, and they will infect everything. So how can the worms resist them? The worms don't have dedicated immune cells, so they don't have T cells and B cells and NK cells. What they do to resist viruses is they use RNAi. They use the very efficient RNAi that they have. And so if a virus replicates, an RNA virus replicates, at one point it will form a double-strand RNA, and this double-strand RNA will be recognized by the RNAi system, because double-strand RNA is suspicious, could be virus, and then it will be chopped up to small RNAs that we call virNAs or antiviral small RNAs, and these small RNAs will then find the viral genome and leads to its destruction. And that's how the worm resists viruses. How can this be enough to protect worms so efficiently against viruses? the hypothesis that I had was, I called it the inherited small RNA vaccine hypothesis. What I thought was that antiviral small RNAs, these VRNAs, just like artificial synthetic small RNAs, would also be shuttling to the next generations to protect them even before they are born. So they will have this repertoire, this knowledge of the viruses, which allows them to resist them. This was my naive hypothesis. And at the time, there wasn't any natural virus that infects C. elegans. However, people showed that you can infect C. elegans with different artificial viruses, like a flockhouse virus, which is a very simple RNA virus. And they created a flockhouse virus that has a GFP in it, so you can know when the virus replicates and propagates. It's very easy to follow. And what they found, and I showed it also, is that while wild-type worms, if you infect a wild-type worm that has a functional RNAi system, that it would destroy the virus very efficiently. And as you can see on the left, it's hard to see the worms there, but there are worms that are just not fluorescing because they destroy the virus very efficiently. However, on the right, these are RNAi mutants. Different RNAi mutants can be show the same thing. And since they can't form these small RNAs that will protect them, the virus replicates and the worm shining green. You can also, from the wild-type worms, you can also sequence small RNAs that tile the viral genome, these antiviral small RNAs. So I thought I could use this system, these fluorescent viruses, to see whether small RNAs transfer transgenerationally and has physiological relevance. And can they mediate transgenerational immunity? So I did these very simple experiments. I took these worms that have a functional RNAi system, and I infected them with a virus. I expressed the virus in them, and they destroy the virus, okay, as I just showed you. But then I let these worms cross with RNAi mutants, and I isolated homozygous RNAi mutants. So these worms, the descendants, don't have a functional RNAi system. So just like in the previous slide, when you challenge them with the virus, they should be green, right? Because they can't form their own small RNAs. That is unless they inherit small RNAs from their parents that can protect them, in which case they will stay black. So it's a very clear, simple experiments to do. And what I found was, when I did this experiment, is that 100% of the viruses of the worms silence the virus. Not only that, the effect moves the worms and also self-fertilize their hermaphrodites. So you can also examine their descendants, and you see that their descendants also, they all silence the virus very efficiently. We also identified, and this can last for multiple generations, we also identified in this paper a need for a gene that's called RRF1. RRF1 is an RNA-dependent RNA polymerase. So it's a protein that synthesizes RNA using RNA as template. Okay? Unlike other polymerases that use DNA as template, this is a gene that uses a protein that uses RNA as template, which is needed for the amplification of the small RNAs in every generation, so they won't dilute. If you infect ones that are defective in this gene, then the effect is not inherited efficiently. It stops very quickly. This shows that the RNAs that are inherited are amplified. And the worm's genome encodes for 4 different RNA-dependent RNA polymerases. And I'll discuss this more later on. In the same paper, we also sequenced for the first time heritable small RNAs. We saw that we can sequence small RNAs from worms that don't have the capacity to generate these small RNAs de novo. So this was the first demonstration that small RNAs are actually the stuff that gets inherited. I told you that people knew that RNAi is inherited transgenerationally, but that's the phenotype. It wasn't clear what's being inherited. Is it the double-stranded RNA, the small RNAs, or perhaps chromatin modifications? Because in C. elegans, small RNAs go into the nucleus where they guide chromatin modifiers to the DNA of the target, and it wasn't clear what's being inherited. Is it the chromatin marks or the small RNAs? So here we showed for the first time that it's actually small RNAs that get inherited. And we know a lot more about this now. We also saw that the effect doesn't segregate in the population in a lineage according to Mendel's laws. So doesn't exhibit the Mendelian ratios. It can't be explained just by segregation of the chromosomes. There are different rules. Can you see my presentation fine now? Is it okay? Yeah, good. Excellent.
All good.
Yeah, thanks. Okay, while we worked, in my paper, I worked with an artificial virus, but later on, Craig Mello, the person who got the Nobel Prize for RNAi, showed that this also happens with more natural infections with other viruses, and that the effect also transfer transgenerational in these viruses. So it seems to be a general principle that worms can form this transgenerational immunity against different viruses. We think that it's not only viruses that get silenced transgenerationally. Other traits and other memories can transfer between generations. And after I studied the antiviral immunity, I wanted to study whether different diets can change the next generations. Particularly, I wanted to focus on starvation, because starvation has been studied. The transgenerational effects or the epigenetic effects of starvation have been studied in different rodent models and also in humans. So the most famous study about epigenetic inheritance in humans was conducted using was a big epidemiological study done on the descendants of people that were starved during the Dutch famine or the hunger winter. In the Second World War, the Nazis starved a large population in the Netherlands, and people studied their descendants. And they saw that the descendants of starved women suffer more from starvation and also from other diseases and effects such as schizophrenia, even neuronal effects. However, in these studies, people mostly focused on intergenerational effects, not transgenerational effects. And I'll explain the difference. A transgenerational effect is an effect that affects the next generation, even when the next generations are not exposed to the original trigger, starvation in this case. An intergenerational effect is something different. For example, in the case of the Dutch famine, the women that were starved, Audrey Hepburn was also there as a kid. This is why one of the reasons it's famous. The women that were starved already they were already pregnant. So their babies were starved also directly. So it's not really an inherited effect. The babies are affected. And since the babies already have a germline, you can say that even the grandchildren are directly affected, okay? Only if you examine the grandchildren, you can say that there's a true transgenerational effect that necessitates the existence of a different inheritance mechanism, something unusual. So when we studied C. elegans, we went immediately to the F3, to the grandchildren, to make sure that they are not affected directly by the starvation. And in C. elegans, another advantage is that it's very easy to separate nurture from nature. Starvation is a complicated thing, but the worms, if the worms hatch in the absence of food, then they have a very dedicated program for dealing with the starvation. The worms in their natural ecology probably move between periods of feast or famine. So they find food and then they eat it for a few generations, let's say an apple, and then they consume the apple, and then there will be, for multiple generations, then they will be starved for multiple generations. And if the worms hatch in the absence of food on the plate, even after 15 minutes, they will change the expression of 27% of their genes. So it's a really dramatic genome-wide effect. And 27% of the genes is more than the number of genes that would change throughout the entire course of development. So it's a very dedicated system which has dramatic effects. And you know exactly what you're doing in C. elegans because you can starve them and control their environment in a very precise way, which is very important when you're dealing with complicated behaviors. So we starved the worms. What we did is we took parents, well-fed parents, that were well-fed for multiple generations, and then we split them and we put half of them on food is bacteria, and half of them let them hatch in the absence of food. And then we sequence their small RNAs and mRNAs, and we make sure that we starve them for just 6 days, so they all survive. We don't want to introduce selection into the equation, because the worms can easily survive a 6-day starvation. After 10 days, they start dying. We don't want to select the most resistant ones. So we start them for 6 days, and then we sequence mRNAs and small RNAs, and then we let them lay eggs, and we bring them out of starvation, we feed them, and we let them lay eggs on new plates, and then we pass the progeny to new plates, and we follow 3 generations, and we sequence small RNAs and mRNAs from every generation. And we want to see what happens to the transcriptome and the small RNA pool of the descendants. And what we found was that even 3 generations down the road, the small RNA pools of the descendants reflect the small RNA pools of the ancestors. So if they starve, you see a particular signature, which and you see that the changes in small RNAs and the genes that they regulate are such that the heritable small RNAs are enriched in small RNAs that regulate nutrition-related genes. And that all of this inheritance depends on the presence of genes that are necessary for transgenerational inheritance. So for example, in HRD1 mutants, HRD1 stands for heritable RNA deficient 1. This is one of the many Argonaut proteins that carry small RNAs that C. elegans has. In these mutants, the inheritance is canceled, it doesn't happen. There are also physiological consequences. So there are phenotypic effects to this starvation on the descendants. The descendants live longer, and other groups have shown that they also are more resistant for additional rounds of starvation, and the developmental timing is shifted. So it really affects the next generations. Over the years, so we studied the viral exposure and starvation. Other people studied this as well, but we also, and other people showed that other environments can lead to transgenerational changes. So if you put the worms in high temperature, it leaves a trace on the progeny. And also, this is not our studies, but people recently showed that if you expose worms to different pathogenic bacteria, then they also learn to resist these bacteria in the next generations. And this is all connected to small RNA inheritance. Not every environment leads to transgenerational changes. I mean, we don't know exactly what defines the environmental changes or the stresses that do this, but we also describe certain environmental changes which are very dramatic, but nevertheless do not affect the progeny. Okay? And what affects and what doesn't affect the progeny, this is still mysterious, and we have some clues, but it's complicated. We don't know the answer yet. The most interesting question, in my opinion, and normally when people think about memory, you think about the memory that's encoded in the nervous system. And I think that it's especially interesting to think whether or to examine whether the nervous system or memories that are made in the nervous system can transfer to the next generation for the following reason. So every tissue that can produce a transgenerational effect is interesting, whether it's the gut or whatever. But the brain is special because, first of all, the brain is unique in its ability to coordinate internal and external changes throughout the body. But more importantly, the brain knows how to plan and think ahead and do complicated computations. So it's quite amazing to think about the possibility that the worms can use their brains to plan the fate of the next generations. And this is very controversial also because it kind of goes around this teleological trap that Lamarck and others fell into. Okay, so this is very important. And very recently, just a month ago, I published a theoretical paper about what type of brain activity could perhaps transmit transgenerationally and what can't, and in which organisms. But we didn't only speculate, we also studied this in the lab. But before I'll discuss our work on brain inheritance, I want to discuss the history a little bit, just in one slide or two slides. In the 70s and 80s, there was very controversial research done on memory transfer between individuals and generations. And this memory ended in disgrace. It was a huge headache. And this started with the work of this person, James McConnell, who was a researcher who studied planaria. These are these flatworms, very different from C. elegans. So millions of years apart in evolution. And what McConnell said, so planaria, if you know them, you know that they are the champions of regeneration. They are amazing. They have amazing regenerative capacity. So, for example, they have these complicated brains, much more complicated than C. elegans's brains. They're with lobes and everything, and you can chop them apart. You can chop off their heads, and the part of the tail will grow a new head, and the part of the head will grow a new tail after a few days. You can even chop them to hundreds of pieces, and each of them will regenerate a new worm. So this is quite amazing. What McConnell said was that he can teach these planaria certain things, certain memories, very for example, like navigate a maze and things like this. And then he can chop off their heads, removing their brains completely. And with the part of the tail will regenerate a new brain, it will still remember what the previous brain learned. Okay, this was very, very controversial. And some people replicated, some people didn't replicate it. It was a big thing. However, the fact that people didn't replicate his study was the least of his concern, because McConnell got exploded when he received a bomb from the terrorist known as the Unabomber, who targeted scientists and other people in technology. And this is how his career ended. He didn't die, but he was injured in this attack. Nevertheless, some people continue these studies and study this in planaria and also in other, even in rodents, suggesting that some memories can be transferred between individuals or across generations, but this ended in a disgrace. People didn't believe it, and it wasn't replicated. Very recently, just a few years ago, the lab of Mike Levine from Boston, from Tufts, replicated some of McConnell's experiments. They used a very sophisticated system for automatically tracking the worms, cutting their heads, and monitoring their behaviors. And they said that they can replicate some of these transgenerational, or it's hard to talk about generations in C. elegans, but these effects that persist to the new brain. So perhaps there's nevertheless something there. However, McConnell also said something more controversial than just chopping of heads. McConnell said that he can take the brains of take the planaria that were trained, chop them, and feed them to other planaria that are naive, and this would transfer the effect. Not only that, he said that he can take this, the worms that hold the memory, grind them, and separate them to different fractions. So just the DNA fraction, the RNA fraction, the protein fraction, and he said that what transfers the information is the RNA fraction. And this is quite amazing because this was before we knew about regulatory RNAs. And this idea that you can just feed RNA to other organisms to transmit memory, which ended in a disgrace, I remind you, reached the general population and became very popular. So you can see there's a comic book about it. There's a Star Trek book episode about it from 1984, where they said, you can read the captions that you can feed that RNA is that memory is encoded in chains of RNA. This all ended in nothing, but it's very interesting to study. And if you think about it, C. elegans can be fed with RNA. As I told you, this is standard to have transgenerational effects. So it is very interesting. And we started studying planaria now to see whether nevertheless there's something there as well. But this is still very preliminary. What I will tell you about is our studies in worms, in C. elegans, which is a much more trackable genetic system for studying these type of questions. It's hard to do genetics in planaria. What we did in C. elegans, and we published this, is that we took RNA mutants, so worms that don't have RNA, the capacity to produce a certain population of endogenous small RNAs. They are called RD4, these mutants. RD4 is a double-strand RNA binding protein necessary for the creation of certain small RNAs, endogenous small RNAs. And we rescued RD4's activity specifically in neurons. So only the nervous system can now produce these small RNAs that RD4 produces. And we validated using literally 6 different techniques that RD4 is expressed just in the nervous system and not in the germline. And then we let these worms generate progeny, and we isolated progeny that lost the rescuing array. So the progeny are again RD4 minus. And we sequence small RNAs from them to see whether we can find RD4 dependent small RNAs that were made in the ancestor's brain. And we did identify such small RNAs, and their inheritance depends on the presence of, on HRD1, this one argonaut that physically carries small RNAs in the germline. There are many more mechanistic studies in this paper, just very briefly because it's published. But we weren't after just, making long list of small RNAs. We wanted to see what they do and can they transmit memory and behaviors to the next generation, because that's the most interesting function of the nervous system. So we followed the targets of these heritable small RNAs, and we found that some of them can affect behavior. So what we did is while studying RD4 animals, we found these mutants, these RNA mutants, are defective in chemotaxis. They can't find food properly, but they are defective in chemotaxis only in 25 degrees. In 20 degrees, they are okay. But at slightly elevated temperature, which are still normal temperature for C. elegans, so they are still fertile and everything, they can't find food. However, if the RD4 mutants descend from heterozygous parents, or even from heterozygous grandparents, then their chemotaxis capacity is much improved, which means that and it's enough for the grandparents to have RD4 expressed just in the nervous system for the wild type descendants to have better chemotaxis, which means that the nervous system, using small RNA production, can control the behavior, the chemotaxis capacity of the next generations. Following the small RNAs that are transgenerationally transmitted, we identified one gene called SAGE2, which is transgenerationally regulated to control this chemotaxis capacity. And we show that if you take in RD4 mutants, it is overexpressed because it's not silenced by the brain. So if you take RD4 mutants that are defective in chemotaxis and you using CRISPR, you remove SAGE2, then the worms behave better. They find food better. And it's very interesting because SAGE2 is a germline expressed gene. And in fact, in this paper, we found that the small RNAs that are made in the nervous system control germline gene expression and not the nervous system gene expression in the next generation. So there's sort of a reverse Weismann barrier. The effects move from the nervous system to the germline, but not back to the nervous system. You affect behavior because the germline controls the nervous system in different ways by secreting different compounds and also by affecting the development of the nervous system. But I won't get into this too much. We can talk about it later. The last thing that I want to show you in the last 5 minutes of the talk is that this inheritance, which is very unusual, it's very different from genetic inheritance, and it's not explained by the rules of genetics that were identified by Mendel more than 150 years ago. When we started, people didn't know whether the epigenetic effects actually, the epigenetic inheritance actually has any rules, or alternatively, this is just a passive dilution or transmission of the effects that the parents make. So we wanted to study this. And indeed, in the beginning, people thought that what happens is that a worm produces a certain small RNA that has many molecules of these small RNAs. And as generations pass, the RNA that the parents made just gets diluted until it's gone. And then the effect stops. This was the most naive hypothesis. And people found that typically when you silence a germline gene with double-strand RNA, the effect would last for 3 to 5 generations, and then it would stop. They even call this the bottleneck of the RNA inheritance, but it's important to stress that the effect lasts 3 to 5 generations at the average, at the population level. At the individual levels, that some individuals are silencing for longer, okay? We knew that it can't just be blending inheritance. It can't just be dilution. I remind you that also in the early days of genetics, people thought that genetics works by dilution. They thought that you just mix the genetic materials of the two parents. They called it, this is called blending inheritance. Later, Mendel found that the genes are particulate, they don't blend. Okay? But people for many years thought in blending inheritance terms, also when it comes to regular genetics. We knew that it can't just be blending because every worm produces something like 250 babies. So the dilution factor is enormous. After 4 generations, and these effects last 4 generations, the dilution factor would be 250 times 250 times 250, which adds up to 4 billions, which is completely homeopathic and could never work. Okay. Instead, I told you this in the beginning of the talk, we know that small RNAs amplify. We found that inheritance of small RNAs depend on these RNA-dependent RNA polymerases, which amplify the small RNAs in every generation. And so instead of asking why does it last for just 3 to 5 generations, we should ask why does it ever stop? Okay, and this is how C. elegans small RNAs work. You have different types of primary small RNAs. This could be exogenous small RNAs, it could be endogenous siRNAs or piRNAs or ribosomally derived small RNAs. There are different types of primary small RNAs. They do not lead directly to silencing. What they do is they recruit RNA-dependent RNA polymerases to the mRNA template. And this complex, the RDRP complex, amplifies much more abundant small RNAs, which are called secondary small RNAs. And they are antisense. They tie the entire length of the gene, but just the exons. So they are made after splicing. And they are 22 nucleotides long and start with a G. So why do the worms forget after 4 generations if they can amplify the small RNAs in every generation? we think that they forget because it's not adaptive to remember everything that your parents did. You want to only maintain the relevant information. And we identified in this paper, the Rechavi paper at 2016, that there's an active mechanism that we call the Transgenerational Timer that shuts down inheritance after a few generations. And this timer is made up of proteins that are encoded by genes that we named MOTEC genes. MOTEC means sweetheart in Hebrew, but no one knows this. But the acronym is Modified Transgenerational Epigenetic Inheritance. And if you mutate MOTEC genes, then you get different periods of inheritance. Then instead of lasting 4 generations, there are MOTEC genes where it lasts hundreds of generations or never stops. It's totally stable because the mechanism for resetting doesn't work. Very recently, we also found that while typically an RNA response lasts 4 to 5 generations, approximately, if you stress the progeny, if you start an RNA response, let's say you silence a GFP, and then you stress the F1 progeny using a number of different stresses, either starvation or high temperature or high salt, then the inheritance stops immediately. So stress can reset ancestral RNA responses. And this is not a passive thing. So it depends on the worm knowing that it's being stressed and on stress signaling. Stress signaling mutants don't reset RNA inheritance after stress. Because this is a plastic thing. If the worm realizes that the environmental conditions change, it can change its strategy. This is all connected to chromatin modification. So these small RNAs go into the nucleus. It depends on nuclear RNA factors. And in the nucleus, they guide chromatin modifiers to the gene and lead to the deposition of certain chromatin marks. What mostly has been studied is suppressive chromatin marks, such as H3K9 trimethylation and H3K27 trimethylation. And we found that certain modifications are required for the silent transgenerational silencing of some genes, while others are not. And moreover, this one methyltransferase, MET2, which depositive mono and dimethylation, is required for resetting of transgenerational inheritance. So in MET2 mutants, RNA responses last indefinitely. They are totally stable, lasting hundreds of generations. The sorting of the small RNAs and the decision of whether a particular gene will be silenced or not transgenerationally is made in these liquid-like germ granules, these condensates. They reside outside of the nucleus. And as an mRNA exits the nuclear pore, it can get captured by these nuclear germ granules, and where all the machinery is there. So in these germ granules, you have the RNA-dependent RNA polymerases and many of the argonauts, and a gene can be targeted for silencing there. And if you disrupt the composition of the germ granules using stress or mutants, you get transgenerational effects that are very different. You get ectopic silencing of genes that shouldn't be silenced, and these effects can last tens of generations, and they show a parent of origin effect. So it matters whether you disrupt the germ granules in the father or mother. And lastly, we see that there's variability in the inheritance between isogenic worms, between worms that have the same genome. If you start a response, some of the progeny will silence RNA very strongly, while others won't. And this is something that I observed already in 2011. I didn't know why this is the case. We studied it recently, and we published it just last year, where normally when you do an RNA experiment, you silence genes, you feed them with double-stranded RNA, and then in every generation, just select a few worms to propagate the next generation. In this paper, what Leah, my student, Leah Rechavi, did is that she followed thousands of individual worms in a lineage. And she tracked more than 20,000 individual worms to really see what are the segregation, how does the segregation of the effect looks in the population, in the lineage. And she found, and we looked for ratios, just like Mendelian ratios, the 3-to-1 ratio and so on, but we found different rules. We find 3 different principles that described how an RNA response will segregate in the population. And I won't get into this because it's published, but we can now predict which worms will inherit and for how long. And this is not true only for RNA responses that start with double-stranded RNA. It also allows you to predict which worms will inherit the effect of starvation, of stress. And we found that there's a mechanism that depends on HSF1, this master regulator of stress, that generates variability intentionally within genetically identical worms, just as a sort, probably as a sort of a batch hedging mechanism, just like there are mechanisms for creating genetic variability. because you need variability for evolution. And this is the last topic that I will discuss very, very briefly. If you start an RNA response and it gives the progeny an advantage, and at the end of the day, you need to, so the progeny it's more adaptive. The worms make more progeny. But then you ask yourself, are these descendants different genetically in their genome from the descendants of unstressed worms? Because the worms are isogenic, they self-fertilize. They make more progeny, but are they different or not? This is very important because if you want to affect the process of evolution, you need to have something stable. You need a different, to induce a genetic change. I think this is a very, very important subject. We found very recently, we pre-printed it just a few months ago, the paper is under review, that the progeny are different genetically. And this happens because of an indirect effect. What happens is that if you stress worms at high temperatures, this affects the production of small RNAs that affect the functions of the sperm. And when the sperm doesn't fertilize the egg in the hermaphrodite needs sperm. It needs functional sperm. So when it senses that it's not being fertilized, it secretes a pheromone that would attract males. And then the males fertilize the eggs using their own sperm, and the worm can propagate. This affects transfer transgenerationally also to unstressed worms. And the inheritance depends on small RNAs because we have a background tagged HRD1, this argonaut that we can deplete, and it stops the inheritance completely. And we showed that when you stress worms, because the progeny mates more, it has a much more diverse genome, because when you mate, you dilute your genome in half, which means that indirectly small RNA inheritance can control genetic variability, giving evolution more raw material to choose from, affecting evolution. Then we did lab evolution experiments to show this. So just to summarize, people often contrast Lamarck with Darwin. Okay? However, in fact, Darwin believed in the inheritance of acquired traits. Okay? Even wrote about it in The Origin of the Species, version 6. He had a wrong mechanism of inheritance. It's called pangenesis, where he suggested that every cell in the body secretes gamules, or these vesicles, that collect in the embryo to constitute, to vote on the constitution of the offspring. Lamarck and Darwin, both of them didn't know genetics at all, and both of them believed in the inheritance of acquired traits. And recently, people suggested that perhaps heritable small RNAs are equivalent or analogous to Darwin's gamules. And this is it. I'll just say that we think that in addition to DNA, which is obviously the main molecule of inheritance, RNA was always there in the background, affecting the inheritance of many different traits, at least in C. elegans, perhaps also in other organisms. We can discuss this. And when people sequenced the genome 2001, they were, of course, surprised to see that humans and worms have the same number of genes pretty much, and some organisms that appear to be simpler have more genes. And so it's not how many genes you have, it's how you control them. And perhaps heritable small RNAs or epigenetic information can affect the control of genes and explain the inheritance of traits transgenerationally. So this is it. And I'd just like to thank everyone in the lab who actually did the work, and you for listening. Thank you very much.
That was incredible, Oded. Like it's such a fascinating talk. This was so
Thank you.
Amazing, absolutely amazing.
Very much.
Yeah. Is it the only problem with Zoom talks is that you can't hear what's inevitably a massive round of applause from everybody that's online.
Thank you very much.
It's just amazing, absolutely amazing. I was just thinking I didn't do my degree in human genetics all that long ago, but everything has changed utterly in that period of time. So that is just mind-blowing, really. I was just going to say to Fernando, if people would like to put questions in the chat, maybe you could triage and invite them.
Absolutely. We do have a question from Massimo. Did you check whether morphology or cell markers of the chemoreceptor neurons change? And is it transmitted in those animals with transgenerational chemosensory defects? For example, did you check on the AW cough -AW-CON neurons morphology and markers?
Yeah, so this is a great question. Do the smaller do the neurons actually change transgenerationally or not? You can do this in, for example, when it comes to AWC neurons, which are important for the chemotaxis of the sensory neurons that actually mediate chemotaxis to the odors that we examined in our paper. Are the neurons changed transgenerationally? It's a difficult question. I'll just say that in our paper, we also did calcium imaging using microfluidics chips to see where the AWC neurons of the progeny are functional. And we see that they are functional. So we don't think that the defect is then necessarily at the level of the neurons. In fact, we think that something else is happening. We think, and I briefly mentioned it, that the small RNAs that are made in the nervous system affect the germline, and they can either do it directly by moving themselves, or perhaps, this is hard to exclude, they might change other secreted factors that transfer from the neurons to the germline. The bottom line is that the germline small RNA pool is changed, leading to transgenerational changes. In the next generations, when we look at gene expression changes, and we also, when we do single molecule fish to track the genes that change, we see that the genes that change are mostly germline genes. Somatic genes get re-expressed normally. So it's sort of a reverse Weismann barrier. The soma of the parents connects with the germline, but the germline of the parents doesn't connect with the neurons. So how is behavior affected if you just affect germline gene expression? We think that it is affected because changing germline physiology affects other somatic tissues. For example, in worms, if you just ablate, these are